So good morning, everyone. Thank you for coming here in an early morning on a Saturday, and we really welcome you to be here to attend our the Applied Knowledge Exchange session. This one is called Bioinformatics Software Testing and Quality Assurance, and I'm really excited that so many of you have registered, and because this is a fairly new topic, we realize this kind of topic has not been featured in any previous ISMB session, so we're really excited that a lot of you turn up and decided that this is a topic you would want to learn a bit more about. So my name is Joshua Ho. I come from Australia in the Victor Chang Cardiac Research Institute. So today we have, I think, a very good combination of speakers. We have statistician, I'm a bioinformatician, we have a clinician here as well. So hopefully give you a sense of bioinformatics, software testing, and quality assurance from various perspectives. So let's go ahead. So this is the kind of the introduction. So let me read in case you can't. So how do I know I didn't make a mistake in my computer code, Professor Smith? Did you double check it? Yes. Triple checked it? Yes. Quadruple checked it? Yes. Did you confirm a hypothesis? Yes. Then stop checking it. But I think, humor aside, this is a big question. So how do you know your computer code is actually correct? This is a big question. So, as bioinformaticians, this is what we do every single day. We write computer code, or we are using someone else's computer code. How do we know it's correct? What does it mean by checking it? How many times do you check it? When do you decide to stop checking it? That's the kind of question we will be touching base today. It's a fairly large topic, so software testing itself is a fairly large topic if you want to do it comprehensively. But today, hopefully we'll get an introduction to what that might mean. So this is actually a topic that is, actually it's a very interesting topic. A lot of people are talking about it and thinking about it. And these are just several examples of, high-profile news articles. So these are not research articles, but they are news articles that are feature articles in Nature and Science talking about various problems. Back in 2010, there are already discussion about, computational errors and what that might mean in science, troubling trends in scientific software use. That's published in Science in 2013. The next one, Mozilla plans to seeks to debug scientific code, 2013. And this year, I think just several issues ago in Nature, it is publishing an article on no more hidden solutions in bioinformatics. And it's about reproducibility. It's about if we are trying to use bioinformatics software in precision medicine, personalized medicine, or whatever medicine you like to call it, if we want to use bioinformatics to analyze human genome data and to provide clinically relevant information, there are something that we need to do. So this is a survey. It's also published in that Nature article in 2010. It's a report of a survey. I'm not sure whether you can see it properly, but the whole idea here is this is a survey of 2,000 scientists. Many more scientists now than before. In fact, about 45% of the scientists are spending more time developing software than before. And a very large number of them are interested in learning more about software testing, but not a lot of them know about software testing enough to call them, having expertise in this area. So that's the kind of situation we are dealing with here. So there are, before we go on, there are some definitions that I want to make sure. No, but the guy said he is capturing the slides as well. Okay, sure. So there's some definitions to help us, go through the slides. So these are some words that will occur, very frequently throughout the session. So might as well explain them now. So error. Here I define as a defect in a human thought process due to misspecification of, for example, misspecification of range of variable. So it's about human error. So all computer errors come from human, right? So we make mistakes, a programmer make mistakes. So this is something that we introduce. A fault is basically what you what is happening in the code or in the way you put together the pipeline, for example. These are concrete manifestation of an error within the software. Could be a bug, wrong use of parameters, incorrect software dependency. Could be all of that. A failure is the departure of the operational software system behavior from user expectation. So it's actually the outcome. After you execute the program, you observe a failure. What is test case? Test case is really the input, the input of the program and a set of execution conditions that is developed specifically to verify the compliance of a program to specific requirements. So it's the input of the program. The last one is an oracle. An oracle in this case is a mechanism to check the correctness of a test result from any given test case. So that means we are not dealing with just giving one input. We want to know that one input is correct, not only for a small number of input, but for any input. So this is an oracle. It's a mechanism to systematically determine the correctness of the output. So you'll see those terms frequently in this session. Why is software testing challenging? It is challenging for several reasons. It lacks rigorous review compared to, say, journal peer review process. We do not spend enough time and energy on testing. Sometimes we misuse external software components. So we are using an R package, or we are using someone else's code, or maybe the software was developed correctly, but we are using it wrong for the wrong purpose. We think it is doing A, but it's actually only doing B. So we're misusing a piece of software. We rely mainly on validation testing, which is merely checking if the result makes sense. So that means you look at this, the value kind of makes sense. It is within the range of expectation, but not knowing that your range of expectation is actually quite large. So that's something we have to deal with. And this is because oftentimes it is actually very hard to check the correctness of the output exactly. Especially if you have a complex algorithm or your input or output is huge. It's actually a big problem. We often rely on very small number of simple test cases. So, to determine whether a piece of software is correct, we typically take what the developer provide, the example code, for example, run it, or maybe come up with a small number of test cases ourselves, run it. If the result kind of makes sense, we generally stops. And I think that's a problem. You'll see later today, after the morning break, we'll actually do a practical session together. We'll actually, take a program, we'll actually try to test it, and hopefully we'll see that, small number of test cases actually you may not be revealing all the software failure. So we'll actually do an exercise on that. Okay, so here is just an example. You have installed a new R package. Maybe this is a package that was published in a paper, you've seen it, and there's a help page in R, and you can see it gives you some general information about the field, what the program is supposed to do, the set of input parameters, the output value, and some example code. So is running example code alone sufficient? If not, how many test cases do we need? How to generate additional test cases? How do we verify the correctness of the output? These are all the kind of things we're going to describe today. In particular, we will have a lot of focus on genomic medicine, because this is an area that a lot of people I think, would be interested in. And also, nowadays, not only genome analysis is no longer restricted to research, it's kind of starting to move into clinical application. Precision medicine, personalized medicine. These are, very buzzword nowadays. And we really, and quality assurance become a much more prevalent problem. So this is a typical pipeline. This is one of the many pipelines that you can identify. It typically starts with the raw data in FASTQ format, perhaps. You align the FASTQ file. You go through a variant calling pipeline such as GATK, for example, where you do a lot of alignment correction and do variant calling. At the end, you annotate your variant calling file, and you end up with an annotated VCF file. So it started with a very big FASTQ file, you end up with a smaller but still fairly large VCF file. The question is, how can you have confidence that the output VCF file is indeed correct with respect to the software that you use or what you think it is doing? Sometimes it is easy to check whether you have a false positive. For example, you can do, once you say you have identified a variant, you can do Sanger sequencing to determine whether that is actually correct. The variant is indeed there. But false negative, on the other hand, is much harder to identify. False negative in this case is you actually have a mutation is in this particular locus, but because the software did not find it, you're missing it. And if you're missing it, it's actually very hard to find it and to do any kind of validation afterwards. So false positive is we can identify. False negative is really hard to identify in this type of pipeline. And this is highlighted by some of the papers that was published 1 or 2 years ago. So these are both papers in genome medicine highlighting that even with some of the most commonly widely used programs, they are not very concordant. The concordance is not as high as we think. And also not only in terms of variant calling, but also in transcript annotation, which is an important part of analyzing whole genome sequencing data. So if you have, for example, in the first paper, they are comparing 5 different pipelines, and it showed that their output is not concordant. So in that situation, how do you know which program is correct? Well, they know they're not concordant, so something must be wrong, right? Either all of them are wrong in some way, or at least one of them is wrong. So we need to know which one is wrong, but in what way as well. So they're not completely wrong for all of them, but the concordance is not as high as we expected. So these are all the questions. So there are at least 2 techniques that we are, I suppose, intuitively already using without realizing there is a name. So these are names that are used in the software testing field. So one is called special test cases. So these are test cases where we actually do know the expected outcome, where we do know the expected output, right? You can always construct some test cases, even though in general it's very hard to determine the correctness for every single cases. But for some test cases, for example, where the input is really, really small, where you can manually determine the output, you can do that. But for large input, it might be a lot harder. And N-version programming, that is if you have several versions of a program or several programs that's supposed to implement the same general specification, we can do a comparison. We can check that the output is the same between them, and that's checking for concordance. And this is the N-version programming, which is basically what a lot of us do when we publish a paper. We want to compare, correctness. A new version actually computes the same output from an older version that might be slower, for example. That's the kind of situation. So, and these are techniques that has been used in the bioinformatics field as well. So this is another kind of program, network simulation or biological model simulation. So this is SBMS simulator. People have been developing test suites to test that, which is a collection of test cases where the expected outcome is known. And this one here, it is the comparison of many simulation software. And this makes use of N-version programming. So these are techniques that we already knew somewhat without using those specific names. There are also other techniques, but realizing that these are software testing techniques that we have been using, it is important because that means there are a lot of other techniques in software testing field that we can borrow and use in our work, and we'll discuss that later in the session. Okay, so the main objective for today include understanding the importance of software testing in bioinformatics, understanding basic concepts and techniques in software testing, and understand how we can implement quality assurance in bioinformatics, particularly in clinical genomics applications. So I've just done the first part, welcome and overview, and next we'll hand over to Ruth and Chris, where they will present some of their work about precision cancer medicine and quality assurance and their experience of implementing that in Oxford. And then we'll have Eleni, who will give an overview on how we can implement quality assurance in clinical diagnostic laboratories. And then we have morning tea, and after the morning tea we have a good practical session where we'll actually try to implement some of this in practice. All right, so, and for the practical session you will need R. You will need R. The level of R programming that we require is really, really minimal, but if you have not installed R, let me know, or I'm very happy to help you during the morning tea break, or if you have a question about R, let me know. All right, okay, any questions so far? Okay, so without further ado, let me introduce Ruth. She should come first.